Good evening, everyone. This is Omark Azrak. I am an MD at the University of North Carolina at Chapel Hill, and I work with IBIS. Today, I'm going to present you our latest research project titled Early White Matter Microstructure Alterations in Infants with Down Syndrome. This research project studies Down syndrome, which is also known as trisomy 21. It is the most common genetic cause of intellectual disability, affecting approximately 1 in 600 newborns. The aim of the research project is to study the white matter microstructure of the brain. White matter is the part of the brain that connects between the gray matter to the rest of the body, and its main function is relaying the information between the gray matter and the body. Um, and we are aiming to study the microstructure of the white matter, meaning the changes in white matter that we cannot see with the naked eye. However, we will be able to see through advanced imaging modalities. To our knowledge, no research has been done in the white matter in infants with Down syndrome. Most research has been done in older children and adults. We use what we call diffusion tensor imaging. Diffusion tensor imaging is an imaging modality that is part of the magnetic resonance imaging MRI that will show us the diffusion within these white matter tracts. And through that, we can know the microstructure of the white matter. We also have what we call the neurite orientation dispersion and density imaging, which allow us to distinguish between intraneurite, extraneurite, and CSF compartment. So it's a more advancing technique to show us the white matter microstructure of the brain. So we had 49 infants with DS and 37 typically developing infants. And first, we acquire what we call BWI, diffusion weighted imaging MRI, which gives us, again, the water diffusion inside the brain. From that, we can extract what we know as DTI, and the colors here reflects the white matter tracts, and then we can extract tractography, which are the white matter tracts. And from the tractography, we can get what we call DTI and NOD metrics. These metrics are numbers that will reflect the microstructure of the brain. We have the fractional anisotropy, FA, AD, RD, NDI, ODI. A decrease in the FA as well as in the NDI, usually reflect and suggest a decrease in the structure and integrity of the white matter and decrease in the density of the neurites. Decrease in the AD suggests disruption of the axonal organization. An increase in the RT suggests delayed myelination, and an increase in the ODI suggests dispersion and fanning of the neurites. So we studied 9 tracts in our research project. 3 of them are interhemispheric, meaning tracts that connect the 2 sides of the brain, and 6 that are intrahemispheric, that are within one side of the brain, one hemisphere, and they have different functions. So the corticospinal, for example, is responsible for the motor function, while other tracts like the SLF, IFOF, ILF, and uncinate are responsible for what we call higher functioning, including executive functioning and language development. So we found out that in infants with Down syndrome, FA and NDI were lower in multiple tracts, while AD, RD, and ODI were higher in infants with Down syndrome compared to typically developing infants. To give you a more detailed presentation of our findings, within FA and NDI, this is what we call the violin plots. The blue reflects the typically developing infants, while red reflects infants with Down syndrome. In general, you can see that in tracts that are affected, FA is lower in infants with Down syndrome compared to typically developing infants. Similarly, in NDI. So our research showed distinct patterns of delayed white matter maturation in infants with Down syndrome, as well as delayed myelination and increased dispersion. This is the first study that examines the white matter microstructure in infants with Down syndrome. And this is a very important first step that lay a foundation for future longitudinal studies that follow up these infants, as well as potentially developing targeted clinical intervention. Thank you for your time.